All right, an official good morning and welcome to everyone for our season finale of 2022 Plants, Pests, and Pathogens. It is great to have you all here. I'm Caroline Richardson, Program Assistant with the Master Gardener Program. And today we get the wonderful opportunity to have our team, Matt Bertone and Mike Munster, do their fun wrap-up session for the year. And they have prepared a fun session for us that will test our knowledge and also just be a little bit entertaining, I'm sure, with their game, Critter or Not. So get ready to do some polls. There'll be some polls coming up, and you can participate on those polls on your computer. And keep in mind when the polls pop up, you can always drag that window out of the way if you need to move it so you can still see the question or the critter or the picture. Just drag that poll window and you can still use it if that's helpful to you. Okay, so with that, I will hand it over to Matt, who I believe will begin.
Thank you, Caroline.
Certainly.
And let me get this all ready. All right, hopefully everyone can see this.
That looks terrific.
And so yeah, welcome to our end of the season session. So basically Mike and I are going to run the Critter or Not fun kind of exercise. This is meant to be, these are things that come in that could be confusing or interesting or strange. You may have never seen them, and this is mostly, this is often for entertainment, but afterwards, you may see these things out there, and so you'll have this knowledge to bring with you. And so Mike is going to go over the rules in a second. Then after that, I'm going to talk a little bit about some insects and things and do some bolos, and then we'll turn it over to Mike to do his section on some pathogens and some bolos for them. So Mike, you want to explain?
Of course, my explanation would be harder or easier to understand if I have my microphone on. But excuse me 1 second, I didn't close my door here. We sometimes get carts rumbling down the hall and things, especially when we're doing this. So hello, everyone. Hope you're doing well this morning also and enjoying the fall and had a good Master Gardener College. Our Critter or Not, for those who've been around the PP&P for a while, we just started out as a simple Critter versus Not choice, but we expanded this the last time to be a three-way decision for each case that we're going to be presenting. So the choices that will pop up will be either critter, pathogen, or not. And just so we're all on the same page, when we say critter, we're meaning any kind of insect, mite, spider, so those would be the arthropods, or any kind of a mollusk, like a snail or so forth, or vertebrate animal damage. So that would be, if you see something that fits into one of those categories, you're going to choose critter as your choice. Pathogen will be the choice for any disease-causing organism, whether it be a fungus, bacterium, virus, nematode, plant, parasitic plant, and so forth. And if it's neither one of those two, maybe it's an organism, maybe it's just a beneficial or just decaying dead organic matter, sapro we call them, or maybe it's not even an organism at all. Maybe it's a plant problem due to some kind of environmental stress or poor management, or even a normal plant structure that was mistaken for a problem or genetic abnormality. Any of those you would choose not. So we still call it critter or not, but your choices are now critter, pathogen, or not. And then the final sort of rule is keep track of your score as you go along. And if you are as a part of a group, so if you're sitting in with other master gardeners, then find someone in the room who can be the secretary and decide by vote what answer you will give, and also keep track of the results. All right. Now Matt's driving. He's in the adjacent office here, and I've just got to say next slide when I'm ready for him to do that. So next slide. So this was a recent sample this month, a Japanese boxwood. It was from down in Beaufort County, and you can see these individual branches that are dying on it, and the rest of the shrub looking green. Is this critter pathogen or not? And Matt will pull up the poll there. See, he's already biting his nails. I know, is that a good sign? He's feeling the emotion for you. So you should by now on your screen see a, oh, I know you do because I'm seeing the results pouring in. This
I
is
know,
great.
wow. So we also maybe have to decide how long we want to let people do this for. I don't know if I see the timer keeps resetting, which is interesting. It looks like. Huh. Are you seeing that, Mike?
No, I don't see the timer.
Okay. Okay. So I think I see the timer now. It says 30 up, 37, 38 up. We want to give it a minute, or how long do we want to give people? I think it's evened out pretty well.
Yeah, I think we can just go by the curve as to
Yeah.
once it looks like everyone who wants to vote has done so.
Okay. Oh, 67 of 67, 100% answered. Wow.
But there's
All
92
right.
participants, so I don't know how that
Oh,
works.
that's strange. This is
I
It's
think
fun.
there's 88, I think, folks who could vote. I think they don't include us hosts. So maybe up at the
Okay.
top there, it says 67 of 88 participated. So maybe
Interesting.
that's the number we look at
Yeah, there you go.
and call
Okay.
it when it gets high,
Okay. Well, I think we're good for this one. We'll keep
Oh,
we
yeah.
gotta keep moving on. So let's share these results. We have Mike, you wanna drive this?
I'm not sure that I can
Can you
drive
see?
it. Can
Oh, sorry.
yeah, did
Yeah.
you? So
Yeah,
does
so
everyone
I'm
see the
sharing it.
results? How about this?
Yes.
Does everyone see the results?
Yes, we do.
Okay. We have 15% of people thinking it's critter, 54% thinking it's pathogen, and 31% thinking not. Okay.
All right. Well,
Okay.
I would have been in the majority when this sample first came in, had it not been for the eagle-eyed agent who, when he submitted it, go ahead and click on the next slide there, Matt. When he submitted it, asked me about the specks that were down in the lower stems. And I looked at that and I said, that doesn't look like a speck. And when I peeked it on a microscope, I saw the tail end of one of Matt's friends there. And I would have suspected something like boxwood stem canker, or now called boxwood dieback, or you might think Volatile blight in a situation like this. But it turned out to be neither of those. It was actually the black twig borer, whose scientific name I'll let Matt pronounce.
Yeah, so Xylosandrus compactus, compactus being because it's tiny. Yeah, I looked in the system. I think we had maybe one or two boxwoods. Maybe I think it was only one boxwood ever that we had submitted with black twig borers, and that was before I got here. So not a common host, although black twig borer attacks a lot of hosts. We've even been hearing of in Louisiana, Italian cypress is getting attacked by it. We've had a number of hosts, most commonly magnolia and dogwoods, but they go after a lot of things.
And I believe that's on the next slide, unless you took it out.
I'm not sure. I don't know if you gave me a next slide. Maybe you did, but oh gosh, deja vu. I don't know. I don't think there's a next slide. So the next slide is another one.
Okay.
So,
well, I had meant to, if I didn't, toss in a picture of an example of the dead or flagging branches on the magnolia just outside the building here.
Okay.
So,
No, I didn't see that. Yeah. So, but yeah, everyone, if you check out any flagging branch, especially on magnolias, southern magnolias get hit a lot. Follow it down. Look for a tiny pinhole. You can crack that open. If you find these little kind of brownish black beetles and little white grubs in there, that's probably going to be black twig borer.
And
all right,
you're probably familiar with the drumbeat of these ambrosia beetles not being primary pests. Well, this is an exception. I'm not trying to steal Matt's thunder here, but this is an exception. This is a beetle that will attack healthy plants, although in this case we decided to do a little digging and see if there weren't any stress factors that might have been influencing it. And I believe we did find some Phytophthora root rot or possible Phytophthora root rot on this. So
Yeah,
Um,
they're an interesting species in that they can attack ostensibly healthy plants, but I have, you know, those magnolias, like our magnolia outside is a very healthy plant. But I think in certain situations, a little bit of stress on the plant will cause them to be more susceptible or more attractive to these beetles. So,
And they don't work alone. Apparently, there's a fungus that they introduced that's also participating in causing symptoms, or at least been proven to do so in a couple of different hosts. But it's not the same kind of, we don't think of it in the same categories we would like a Dutch elm disease or a laurel wilt, where you have this aggressive pathogen that's being brought in by the beetle.
I'm seeing questions come in. Let's wait till the end to do that. And I guess we got to keep moving on just because we got a lot of these stuff to cover. Okay, next one. Let's see. All right, so this was on a section of wood in central North Carolina and in April. And so I'm gonna, how do I stop sharing? Let's see. Okay, good or not. Oh my gosh, where's relaunch poll? Okay, I should relaunch that then. So everybody should see that hopefully. So critter or not?
All right, I see the little counter there at the top now, Caroline, with the 53 of 95. And that there's
Yes.
99 total, so I guess we don't have the franchise yet.
Yeah, I wish that it looks like the time is actually working. It just keeps going back down to 0 occasionally. I think it's actually
Yeah.
counting up. So we'll do it when we get okay. So it looks like we've got 70% of people. Let's get, when we get 75%, we'll stop it. Although I don't know, it's leveled out, I think. All right, now we got 75. Okay, I'm going to end the poll and for time's sake and share the results. So we have 59% of people think it's a critter, 21% think it's a pathogen, and 21% think it's not. Is everybody seeing those polls? Yes? No? Okay. I'm going to stop sharing now and let's see. So those who said critter are right. This is the nest of a sweat bee. So these bees, that yellow stuff, that powdery yellow stuff is actually pollen that they've collected. And this group of sweat bees, they create mud nests, mud cells under wood, under bark of dead wood and things like that. Stuff it with pollen, and you can see the little larvae there too that are feeding on that pollen. So this, we've had a couple samples of this. Somebody uncovered a blanket or a carpet that was outside that was kind of just folded up out on their lawn or their ground, and they overturned it and found this too. So they will kind of get into little spots, spaces, and create those mud cells. So good job. Looks like a majority of people got that one. Okay, next one.
So this was a photograph that was sent in, not through the system, but as an email, where the agent had a suspicion of what this was, but wanted confirmation. And the situation is a compost bin. So that piece of lumber that you see there is the border to a compost bin there in Haywood County. The photograph was taken in May. So what do we have? Is it critter or not?
I love watching the numbers go up and down. The bars go up and down.
It's almost like it's one of those meters that you put on when you're listening to music, and it just kind of,
Yeah.
kind of equalizer.
All right, we got 75% of people, but let's give it to 40 seconds. How about that? We got 12 more seconds. Okay, wow, 80% of people. All right, we're gonna stop it there. All right. Mike. So it looks like we've got 10% of people say it's critter, 47% people say it's a pathogen, and 42% say it's not. All right. Drum
Well,
roll,
that's-
please.
Yeah, that's a very interesting result. I'm impressed with the number who said not. And even those who said pathogen could be forgiven in this case because, yeah, this is a fungus, not a plant pathogen though. This is Xylaria polymorpha, and we've talked about different members of this group of fungi in the past. Of course, this is what it would look like in the springtime, but they can last quite a while. And the common name, and it really does look like it when you see this photograph especially, but the common name of the fungus is dead man's fingers. And if you were to excavate this and look, you could trace the fungal structures down to a piece of wood there from which they come. So these are decaying the wood, but they're not going to be causing a problem in that in the garden there.
And they turn darker when they get older, right? They turn kind of black almost.
Yes, I
Or-
believe they do, and then
Yeah.
before they break down.
Yeah.
So-
All right.
so-
Spooky. All right, let's see. And on to the next one. All right, so this is Yopon holly from Brunswick County in July. So what do you think? Let's see. Let's relaunch this poll. All right, critter or not? Well, critter pathogen or not? Those are those are your choices.
And just as a reminder, pathogen is a disease-causing microorganism. So even if you think it's a microbe of some kind, if you don't think it's actually causing plant disease, then you should choose not.
Okay, we got 60, 70%, almost 75. So wait till 40 seconds again and we can end it. All right, I'm going to end the poll, share the results. Okay, we've got a almost unanimous critter at 91%, 8% for pathogen and 1% for not. Okay, so this one is, it is a critter. So a lot of you got it correct. That is webbing on this Japanese, this Yopon holly, and it is from fall webworms. So fall webworms do attack a lot of different plants, especially woody plants, creating this large webbing. And most people notice it on the tips of trees. But this sample was a low shrub, and there were these caterpillars were all over it. Now, fall webworm is pretty diagnostic, pretty easy to identify because it's long hairs. Usually in the south here, I've seen mostly it comes in this whitish yellow form with these black patches. It does come in other color forms around, but they have these very long, seedy. The longest hairs on their body are longer than five of their body segments, and so that's how you can tell that you have fall webworm. Of course, the webbing helps too, but this was a fairly interesting sample. I've not seen them kind of attacking these low bushes very often. So great. Well, good job, everyone.
So Matt, can I ask a question?
Sure.
How would you know that's not a salt marsh caterpillar? Because when I think fuzzy white caterpillar, I think of the salt marsh caterpillar.
Yeah, so a bit smaller. So this is as big as they're going to get. They're only about I want to say about an inch long and kind of thinnish. So they're much smaller than the salt marsh caterpillar typically. Also, that cedal length is really key. So it's got the salt marsh caterpillar. None of those cedi will be as long as or longer than 5 segments. They're all going to be shorter. Very similar. It's actually a close relative though. They're both they're both tiger moths. So that's why they look similar. Tiger moth caterpillars can sometimes be difficult to figure out which one it is. But this one, that pattern, those really long hairs, the size, and kind of that's webbing is pretty characteristic of that. But great. Yeah, good to clarify.
Cool.
All right.
So what we have here is the base of a stem of an American beautyberry. It came from Transylvania County, submitted last month. Now, the problem with this sample was, and I hate to admit this, but I sort of mocked this sample because they sent it in a year after the plant died. And normally that's a no-no because we, whatever the problem was would have been long gone. So in this case, though, it turned out to be quite interesting. So I'll just, without describing them, I'll just let you observe what's in the photograph here and decide critter or not.
All right. Let's see. I love all the participation in the majority of participants.
Yes, this has been great today.
All right, we're at 75%. We'll wait till it's 40 seconds. Okay, you'll get your last votes in real quick. All right, I'm going to end the poll, share the results. All right, looks like we have 12% critter, 54% pathogen, 34% not. Okay, I'm
All
gonna
right,
stop
Matt,
sharing.
if you would do the reveal.
Yes. All right.
So this turned out to actually be a pathogen. Although when you're talking about a year after its demise, it could have been something secondary and a not. But no, in this case, it turned out to be the seldom seen, well, back up. This is a fungus. It's called Rosalinium necatrix, and it causes a disease called white root rot. And this stage of it that you're seeing on the right, those black spherical lumps, that's the sexual stage that produces what are called ascospores, and that's the micrograph on the right there, which is seldom seen actually. That takes a long time to develop. And then there's the other stage, the asexual stage, which is on the right there, that other micrograph on the, I'm sorry, on the left. But the fungus itself is usually not, doesn't get to this stage where you'll be able to identify it. And I've actually not encountered it before, but it happened to be on the sample and is the presumed cause of the death of the American beautyberry there.
All right, thanks, Mike. All right, next one. All right, so this is from a loblolly pine in Randolph County in May of 2021. Relaunch the poll. All right.
And do pay attention when you, I'm sorry, Matt, I'm sorry.
Oh, sorry. No worries.
You have to, it's not official and you say critter or not.
Yeah, critter or not, or pathogen.
No, I just wanted to emphasize the importance of paying attention not just to the photograph, but the location and the date, since some things are going to be regional and other things are going to be seasonal.
Ooh, this is looking like a fun one. How are we going? Let's see. Okay, we got almost, okay, it's neck and neck just about. All right, let's give it 10 more seconds. All right. All righty, I'm going to end the poll, share the results. All right, we've got 31% think it's a critter, 30% think it's a pathogen, and 39% think it's not. So this is a spectacular one because it is completely confusing. So I'm gonna stop sharing and let's move on to the answer. It is
A pathogen. So this is a fungal pathogen. It's one of the needle rust fungi, and certainly we only have the photograph to go on, but it's certainly one of the needle rusts in the genus Coleosporium, which is one of those rusts that alternates hosts, in this case between a pine and almost surely some kind of or some member of the Asteraceae. So it could be Solidago on goldenrod. There's one, I think, not 100% sure. I think there's one that alternates on Vernonia. There may be multiple alternate hosts as well, but this would be a needle rust fungi. So the spore is produced here would, if they're lucky, infect one of the alternate hosts, one of the Asteraceae, and then later on the spore spreaders there would infect the pine. And it's not a serious issue. It's a curiosity, but it's not going to really cause any serious damage to your pine.
I love these double host rusts. They're always so weird. They're always great critter or not. All right, moving on. Let's see.
Double host rust, the official name for that actually is heteroecious, meaning having different homes. Or different houses,
Yes.
different
There
houses.
you go. All right, here you go. Okay, Mike.
So the setup on this one,
Sorry.
oh, he's already
Yeah,
on.
let me end the poll. I'm gonna end the poll real quick. You start it again.
All right, rewind.
Rewind.
These photographs were sent into the system. Not again ideal. That would just be pictures of a single leaf, but maybe that's all that was provided. The good thing was that they took the precaution of photographing both the upper and lower surface. Camellia, Pender County in October last month. So critter, pathogen or not?
All right, so how are we
Okay, yep.
doing? We're at almost 75%, but we're only about 30 seconds in. We'll give it to 40 seconds.
I think 40 seconds is a good time in general.
Yeah.
Not too long, not too short.
All right, looks like we're at the end. I'm going to end the poll and share the results. All right, so we have 73% of folks think it's a critter, 23% think it's a pathogen, and 4% think it's not. All right, I'm going to stop sharing and close this up, and we'll go on to the next slide, which will reveal that.
Yes, in this case, it was the 4% that were correct. So most people thought this was a critter. Matt, how would you explain that this is not a critter?
Yeah, so I'm assuming people thought it might have been T-scale, which is very common on camellia, of course, and looks somewhat superficially similar. But based on, the lack of wax, lack of distinct kind of similar shaped and sized critter or structures, these are all fairly random sizes and shapes. And you can see they're also showing on the top surface, which would not be T-scale. You'd have yellow patches on the top surface, and you'd have a little bit smaller and more discrete, very similar structures, because all the scales would be about the same size and shape.
So in this case, it turns out to be a physiological condition called edema, which is related to water, the water balance in the plant, and it's not entirely clear to me how this happens. We often see it on thick leaf plants, so ligustrum would be another one, geranium, especially the ivy geranium, some others. The old explanation was, and I'm not saying this is outdated, but maybe incomplete, that we had, if you got a situation where the roots were warm and moist, so a lot of pressure from the roots, water coming up, but that the day was cloudy and a poor transpiration condition, so the water was getting forced up into the leaves and wasn't getting transpired away, and so we had the buildup there, which caused the cells to inflate and then later burst, and they become corky at a later stage, kind of like blisters at the beginning. There have been other studies though that have shown that fluctuating water levels, so alternating or fluctuating between wet and dry in the roots causes it to be worse. So I don't have a complete explanation for this. I'll just say that it has to do with the water relations in the plant, and it is called edema, sometimes spelled O-E-D-E-M-A, edema.
I debated whether to put the OE, because I like that one too, but and yeah, this one, they seem kind of like corky or spongy kind of blisters on plants, that, and kind of random sizes, not usually hugely different, but that's you know, when you can't find any critters or pathogens and it looks like that, it's almost always edema, or at least that's the suspicion. So, okay, let's move on to the next one. So this is a coneflower from Wake County in June 2022. And I think you all know the drill. Critter, pathogen, or not?
I think this is the first one. Oh, it took a while, but some did come in with the critter on their choice. It was looking pretty
So I think, yeah,
unbalanced there.
some people have to go against the grain. I think they're like, you know, I'm not gonna answer what everyone else is answering. So
if you like the movie The Music Man, it reminds me of the character of the mayor's wife, and she said, "Oh, I am reticent. Yes, I am reticent."
all right, so we have 78% and 46 seconds. I think we'll stop it there. Let's share the results. Okay, so we have 11% critter, 28% pathogen, and 61% not. Some of you are partially right. So here, let's then this. Okay, this is an interesting one. This is a trick one. This is critter and not. So what we have going on here, these are actually flowers that are outside of our building. And I look at them all the time because they have both these thickened, oh gosh, I don't even know what you call those structures. I always forget on these flowers.
Disc flowers.
The disc flowers are thickened and green and very chunky, but also it's this double flower head and this kind of elongate flower head. And so the elongate flower head is this fasciation, which is basically, Mike, you're going to have to kind of help me with that. It's some kind of physiological or genetic kind of disorder, right? Is it?
Well, it's genetic, but it seems to be, have some kind of environmental trigger as well. And I don't fully understand it myself.
Yeah.
So think of celosia, the coxcomb type celosias instead of the plumose ones. So that's actually a fasciation, but it's a reliable one that you plant the seed and you're going to get it. But sometimes it just shows up. You've probably seen the flattened squash stems and so forth that appear in the garden.
Yep.
So yeah,
yeah. And then these, like I said, these thicker green disc flowers, that's actually caused by the coneflower rosette gall mite, which is yet to be described the species, but it's got a common name, and it's a tiny areophyid mite. I should have included a picture of the actual mite in here, but it's this little tiny yellow mite, worm-like mite that lives in there. They infest the flower head as it's developing, and when they're feeding, it causes this forest of growth for it to feed in, them to feed in. So if you see, this one was fun to put in because it's both, and it's the flowers can get affected by multiple things. And actually, Mike, you think you put a slide in after this, right? So yeah. So if you want to talk about this then real quick.
So some who thought it was a pathogen may have been thinking of the aster yellow's phytoplasma. And this is more of what you would see. You would see the entire, not just part of that flower turning green, but you have the petals there becoming entirely like leaves. So that's called phyllity when it's changed to a leaf-like structure, as well as the virescence, which means that it's green. And this plant was not tested, so we don't know for sure, but yeah, this is pretty typical of aster yellows. I sent a link in the chat to a little bulletin from the Buckeye Yard and Garden Online from Ohio State, where they talk about both the mite and the phytoplasma. You can see photographs of both situations.
And you could have a plant probably with healthy flowers, ones that are fasciated, ones that have the mite, ones that have the phytoplasma. So it's like you could have probably one plant that has all those. I'm sure you could find. In fact, those plants out there, some of them were unaffected by the mites, some of them had the mites, that one had the fasciation. So it's pretty interesting to see like that chimera of diseases and disorders and mites and whatnot. So
But you can get whole plants with the phytoplasma still,
yes.
because it's systemic.
So okay, so let's see. Move on to the next one. I think this is you, Mike.
So this was a, excuse me, let me see here. June of 2020. Yeah, I don't have a great setup on this one. I'll just say that this is a branch of a persimmon that was submitted, and after being cut and put in the humid chamber incubation, there were these strange, round, shiny, milky things there. Some roundish, and then there are also tendril-like translucent things coming out of it. So the question is, critter or not?
Oh, I was gonna say nobody picked not yet, but
You know, I wonder, are we biasing people by commenting on how things are coming in?
that's I don't
This
know.
is-
Well, they should go with their gut. They shouldn't let us. You know, they don't know what we're saying is true or if we're misleading them. So you choose. You know, go with your gut. Go with what you think. Don't let us influence you.
We would never equivocate to try and throw people off, though, would we?
Never. Never.
Not on election day anyway, right?
We got to be very honest. This is one. This is either a critter, a pathogen, or not. I'll tell you that much. All right, so we're at 67%. Okay, it's only about a minute. People are a little hesitant to answer this one. This is interesting. Okay, all right, we're gonna end it there though. So let's share the results. So we've got 33% saying critter, 58% saying pathogen, and 10% saying not. So let's move on to the next slide. Okay. I guess Mike, you want me to take this over since this is kind of mine. This is a critter. This is a super weird thing. Didn't know what it was at first, and actually Swarna Moparthy in our lab, who's the field crops pathologist, but was looking at this sample. She said, "Oh, it looks like there's mites in these tendrils," and said, "Oh," I looked on the scope and said, "Yep." So these are actually what are called physogastric mites. Physogastrics is just a term for any kind of insect or critter that has swollen up, usually with eggs or offspring. And these tiny mites start off like this and run around. And when they mate, after they mated, their body, the female's body, swells up, and inside that swelling are its sons and daughters who then mate and then burst out of her and go and feed on whatever they're going to feed on. Now, this one was very difficult to ID, the actual type of mite. I think it's this Sideropterid, which do feed on plants. But there are some that feed on insects that do this and close relatives, but it was super surprising. And these things were the size of kind of, it's hard to say, but much bigger than you would think a mite would get. Anyway, I was not expecting anybody to really get that. And in fact, this is the first time I had ever seen it. And if I had just seen the photos, the zoomed out photos, I would have never guessed it was mites. But upon further investigation, we see that these are in fact female mites that are just giant. So anyway, weird situation.
And the ones that were kind of stringy were the mites as well, but
yes, that was another.
Yeah,
that was another weird thing. So I think some of the mites had crawled into the vascular bundles, the vascular tubes of the plant. And what happened was, I think when their abdomen was swelling, it kind of just squeezed out, and the mites lined up in a row, and it was kind of like those Play-Doh machines where you kind of squeeze it out. That was my theory, at least. So that made it look kind of strange and stringy rather than these bulbous ones. So anyway, that was a fun one. All right. This next one was from just recently, just the other day in November in Wake County. A Japanese maple was covered in this. You can see the leaves. And we are just going to launch the poll then. Let's see. Critter, pathogen, or not?
Probably we should say that the photo on the left is a magnification
Yes,
of they probably figured that out.
it's the yard where it came from. That's
Drone, there was a
Drone.
drone flying overhead there.
Okay. About 60% of people almost. Okay. All right. Give it a couple more seconds.
Yeah, let
All
me
right.
75%.
Okay, 75%. All right, we're almost
And
there.
we stalled at 74.
Of course, of course. All right, well, I'm gonna, in the interest of time, I'm gonna end it right there. I'll share this results. We've got 18% critter, 75% pathogen, and 8% not. All right. So it is.
Yeah, you folks are too good. And you probably knew that this is one of the powdery mildews. It's one that I haven't seen this well developed before. I've had a suspicion of this before, but not like this, and didn't know what in that previous case what species it was. But after working out in the lab last week and working through a key, it comes out to be the species Sawadea polyphida, and on this Japanese maple. So it is one of the powdery mildew fungi, and what you're seeing there are the structures in which the sexual spores are produced. And it's got these really beautiful branching appendages all around that. It's called a chasmathecium, inside which the sacs of spores are produced.
That's one of my favorite words, chasmathecium. So okay. All right.
All right, I'll give one to you on your birthday.
Okay,
word,
thanks.
The
thanks.
word, I mean.
Yes, I do love how they look too. They're amazing. All right.
And
Mike.
I believe this is our last one, isn't it? Time flies. We have a Bradford pear sapling from a yard in Wake County. And the question is, this off-white sort of column there against that stem and reaching down to the ground or up from the ground, one or the other, is of interest. And the question is it a critter or not?
All right, so let's see. We've got 70% of people after 30 seconds. Some people might be hesitating. I'm not sure. All right, we'll give it a few more seconds.
Yeah, and in the interest of the folks who might be there in a group, taking a vote or whatever, and I'm gonna make sure and give you enough time to be able to sort out your differences. And
all right, we've got 76%. I'm going to end the poll and I'm going to share the results. So we have 21% critter, 48% pathogen, and 31% not. Another kind of equivocal one. All right, I'm going to stop sharing this. All right, so for further context, the person who owned this property contacted me and they were cleaning out their wooded area and found this jar full of moss and said, "Ooh, do you want this jar?" And I do like jars of moss, but I also like jars of moss that have silken tubes in them, because I know from this person that his property is full of purse web spiders. So these spiders, it is a critter. These spiders are a mygalomorph spider, which is tarantulas and trapdoor spiders and their relatives. They're a very strange group of spiders, and they basically what they do is they create a silken tube that goes in the ground and then up a tree. And you can see back here, this is a larger specimen. This is about the size of my thumb. And they sit inside the tube, grabbing on and waiting for an insect to crawl on the tube. at which point they use their gigantic fangs. You can see the chelicerae, these very large chelicerae with large fang there, to rip open the tube and grab the prey and drag it inside the tube. And then they repair the tube and sit inside and wait. One of my favorite spiders ever, and it is now in the lab, and I'm hoping to feed it and keep it and never see it, but maybe see it eat. But I was very happy to see that. So, oh, we have one more. You lied, Mike. We have one more.
Well, this is, we're not actually officially doing this one, or are we? Was this a critter?
I don't know. You included in here, so.
All right, but the count of 12 is going to be important because otherwise the last thing will be, okay, let's say that this is one.
I think that was 12, but we'll
So
see.
this, is this 13 then? No,
I think this
okay,
is number
yes.
12. Mhm.
Oh, it is 12. Okay. So Leyland Cypress, Macon County, September of this year. Christy has been working hard on this situation, and this is one of the photographs that she sent. And of course, the idea here was to, the inspiration, I'll put it that way, to include this was the fact that Matt was doing the purse web spider. Since we have something on the outside of a tree trunk here going on. And
All right,
Yeah.
we're at 71% and almost 40 seconds. Just about a 75%. Come on, one or two more votes. All right, there we go.
Oh,
All right, I'm going to share the results. We have 9% critter, 70% pathogen, and 22% not. Okay. Okay, stop sharing. All right, let's go to the next slide then.
yeah. It is not. No, it's not. So yeah, obviously we're trying to kind of throw you off there by putting it right after the spider, but this is some kind of a fungal fruiting body. So that much is true, that it's an organism, it's a fungus, but it appears to be, it's, well, first of all, this is the most important thing. This is not armillaria. Armillaria will form its mat of mycelium, which will be more of a creamy and white color. It'll be between the bark and the wood. It won't be on the surface like this. And in this case, the wood was not decayed at all. I'm not sure I could say as much about the bark, but I suspect that this is one of the mycorrhizal fungi. You know, usually the mycorrhizal fungi are producing typical mushrooms, you know, like a rustula or an ammonite or something. But there are a couple that will produce this sort of fruiting structure. I didn't find any spores in it. I'm not sure of the identity, but my hunch is that this is one of the mycorrhizal fungi, and it is not the cause of whatever the demise of these particular trees is. All right.
Okay, great. Thanks. All right, I think that's it. So now, now we're going to say, how did you do? So I'm going to launch a poll. Let's see. This is going to be A through E. All right, so how'd you do? So 0 to 2 correct, 3 to 5 correct, 6 to 8, 9 to 11, or perfect 12. And these are anonymous, so feel free to answer. And we'll wait again until we get a majority of people. All right. So.
Not exactly a bell curve there.
Yeah. All right. Let's give it another 10 more seconds or so. I guess people are still tallying, so we'll give it a full minute, let's just say. Okay, almost there. We have almost exactly 2/3. I don't know how far that decimal place goes. But okay, we're going to end it there. And we're going to share the results. So we have 37% of people got 3 to 5 correct, 55% got 6 to 8 correct, 8% got 9 to 11. Apparently no perfects and no 0 or 2s. So that's good. So at least people knew the majority of things. But that's the whole point of this, I guess, is that these are interesting, confusing, kind of strange
Mhm.
things. And so we don't expect you to get perfect. If you got perfect, then you actually come for work for us, I guess. But this is just kind of for fun and entertainment, but also to test your knowledge. And if you happen to see these things later on, maybe you'll say, oh, what the heck is that? And oh, I saw it on Critter or Not. So thanks to everyone for the participation. Mike, do you have any last words for that?
Last words? I mean,
Not last
today.
words.
Yeah.
Yeah,
For the
for today.
season, just
No,
for
for the season,
For the
yes.
for the Critter or Not.
No, this was fun for us, and I hope it was for you too. Yeah, we, like Matt says, these are some of the strange, oddball ones that come along that we like to
Yeah.
toss out there.
We'll hope to do it next year too. So keep finding these weird things, send them to us, and we'll hope to include them. And with that, I'm going to continue on. I'm going to go fairly quickly since we're getting close, but I hope everybody was still kind of engaged and having fun with this. Let's see, we got some chat stuff. Okay, so this isn't official, but Critter or Not, this is on a soybean. No, we're not going to poll or anything. I just want to tell you this isn't Critter or Not. This is actually Critter and Pathogen. But in this time, I'm going to my section today, I'm going to talk to you about some kind of creepy, gross, and weird, or fun, I guess, depending on who you are. Entomopathogenic fungi, so fungus-infected arthropods. So that one was not a pathogen of the plant, but a pathogen of the critters. So fungi that attack arthropods are called entomopathogenic. Arthropods are exposed to spores that penetrate the cuticle. They live inside, feeding on the insect, so the arthropod, sometimes controlling its behavior, as I'll describe in a few slides. And then when the conditions are right, usually high humidity, the fungus produces spores and it fruits out the body, like this very poor long-legged fly on the leaf. You can see all that, those fruiting bodies that are going to spread spores to infect more insects. And so I just want to quickly go through some of these groups, just so if you see them out there in your gardens or in your yards, you're not questioning what it is. So anybody want to guess what type of insect this is? Any question? Any guesses? Type in the chat. So these fungi take a toll. Yeah, it is a dead one. Gene, you are correct. But it is a dead moth. So this is a Cordyceps tuberculata on fruiting out of, you can see these are kind of like the mushrooms, but these are the fruiting bodies of the fungus. And that's what it would have looked like. It's not that species probably, but it's kind of a noctuid moth just sitting on a kind of a rock face. So this is pretty clearly a katydid, but you can see these cordyceps type fungus coming out of the body. This poor katydid was affected before, and it climbed up on this branch, held tight, and died. And that's when the fungus fruits out. It's a common thing to see them up on branches and high up because the fungi want to spread their spores as far as possible. Okay, so any guesses onto what kind of type of organism this is? From this view? No? Okay. Scale? There are fungi. It's a fungus, but what type of organism is attacking? So, or maybe somebody's saying fun. So this is actually another Cordyceps. This is the family that the Cordyceps fungi are in, and this is the Gibellula, which is many of them attack spiders. So this is the one that, so you can see that spider's body there, and this one, another one. These are this fruiting bodies and different stages of this type of fungus that attack spiders. Now, I will say I'm not an expert on fungi. Mike is much more than I am. So I may not be giving you all the details for these, but I think it's just really interesting. Now, there's the Ophiocordyceps, which have the Ophiocordyceps, and those are mainly on ants, but they're on other groups too. And so this is apparently a new species that I took photos of because the species of carpenter ant had not been known to have this. But you can see the different fruiting structures and different carpenter ants. Again, the fungus causes them compels them to climb up branches. They hold on tight. They use their mandibles to latch on, and basically then they die, and this fungus then fruits out and rains down spores on ants nearby. And in fact, when you find them, you often find several ants on one section of branches with these fungi. Here's some other weird little ones. This is probably on some kind of scale insect or whitefly or something that I was told online, but it's got these huge, I forget what the term for that structure was. There was some interesting term. I don't know if Mike knows, yeah, I'll have to find. Mike, you're on mute, by the way.
Yeah, I don't know if there's a special term for it, but it would be a stroma of some kind.
Yeah, okay. And then keep going with these. I don't want to take too long, but and this was a really nice one submitted by Tom Glasgow oh shoot, I'm forgetting the county down the east. It's Mike, I just blanked on
Craven.
it. Craven, that's what it is. So this is a really cool one. This is a paper wasp with the cordyceps fungi coming out of it, and this spider. There's a gall on this oak, and the wasp is now latched down near that, and then there's a spider and its egg sac building a web among that. It's just such an insane photo. I love it. A really great photo showing all these different interactions. Anyway, other cordycipitaceae include Bovaria, which is called the white muscardine disease of insects. It's not as dramatic. The fruiting body is mostly like a white fuzz that comes out of all the joints and body parts. But it's closely related, entomopathogenic fungus. People were asking whether this is a symbiont or beneficial. These are all pathogens. These are all kill the insects. They're parasites of the insects, and they fruit out and kill them. And this goes back to that first picture I showed you. So this was a sample we got in a few weeks ago of nympho kudzu bugs succumbing to Bovaria, this white muscardine disease, on the soybeans. So in the right conditions, this fungus is out there. It's killing pests out there even. Now, green muscardine disease, metarhizium, is a very distantly related fungus, as far as my research has shown, has come. But it produces this green mold-like structures on these insects, and it's often in soil organisms. Now, there's a group called the Entomopterales, and Entomophthora, which really means insect destroyer. If you've heard Phytophthora, that's plant destroyer. So Entomophthora is insect destroyer, and flies often get it. So if you see flies on a branch or plant, they've again gone up to that plant, latched on. This one is even sucking on the plant, kind of to latch on further. And the body just kind of explodes in spores, and you can see all over the plant and raining down on other insects nearby. Here's a soldier beetle that's affected by another Entomopterales, Eriniopsis lampidarum, and so they again latch on, and the spores rain out. And luckily, these entomopathogenic fungi also attack things we don't like, so pests. So spider mites. One of the reasons why they thrive in dry conditions is because they're not affected by this fungus that often keeps them in check, and it kills them. And aphids right here can get affected by fungi as well. Now, another whole different group of fungi that this one's actually sexually transmitted between ladybugs is in this group called, it's one of my favorite groups, the Labobeniales. And this is Hesperomyces virescens. Now, when I took this photo a long time ago, I thought this midge had laid some eggs on this ladybug, but I'd learned that these little yellow structures are actually the fungus fruiting out from the rear end of this ladybug. Now, they will live on, but they're not feeling very good. And in fact, they're on all different types of insects. So here was an ant specimen. There were a bunch of ant specimens submitted, and I noticed that it had all these little weird things on it, and they turned out also to be Labobeniales, Labobenia formicarum. And so insects aren't immune to, no pun intended, aren't immune to fungal diseases. It is fairly common under the right conditions, and you can find lots of insects that are attacked by them. So let's see. I think that's it. I'm gonna just finish up with some bolos real quick. So for late fall and winter, One thing to be on the lookout are fall cankerworms. The adults are now going to hopefully, or not hopefully, but are going to be emerging soon. Now, we haven't had many years recently where we've had large outbreaks, but the wingless females and the winged males are going to be out, the females crawling around and the males mating with them. And the females will lay batches of eggs on buildings, but also on trees on which their larvae are going to feed in the spring when they emerge. There are also a lot of different insects that are only active or mainly active in the winter. Things like winter crane flies might be coming to light, so you really only see them at this time of year. Winter stoneflies and springtails can be active, so it's not everything is going into hibernation. There's some things that really like this time of year. If you're bringing in firewood into the home, be aware that the change in temperature can cause insects that are living inside there to finish their development and pop out. So you may get some wood-boring beetles or wasps that live in the wood that come out. And if you have an influx of these insects after taking in firewood, just know that they're just temporary nuisance. They're not going to infest the home or anything like that. But you did basically bring them inside and rear them out unintentionally. And along those lines, Christmas trees. You know, these are plants that are out in the environment. They have lots of insects and other critters that are on them. When they get cut down, not all of them come off. That squirrel that's in there might jump out, but these aphids and things are not going to do that necessarily. So we often get calls at this when Christmas rolls around, especially after Christmas, when the plants are getting a little drier, of the large or black insects moving away from the trees in high numbers. That's because the aphids now don't have a conducive site to feed and they start to move away. So if you find these this is a little bit lighter, but you'll find these dark formed aphids. They're fairly large and they have these cone-like siphunculæ or the cornicles. That's what's on there. But you may get other types of critters coming off the Christmas trees. Household invaders. You may see an influx of cockroaches or spiders soon, or they're just kind of incidental, or ladybugs and stink bugs coming into the home to overwinter still. But soon enough, once it gets too cold, they're not going to be moving much, and they're going to have already found a place to settle. And finally, there's going to be some opportunistic activity on warm days like we saw the other day. Throughout the winter, you may get flies flying around. They're usually kind of sitting and waiting for days that they can fly around. Sometimes mosquitoes might bite in the winter. You might get some fruit flies or some blowflies around that are winter active. And with that, I'm going to stop sharing to give Mike some time.
All right, thanks, Matt. Do you want to take your questions now, or you're on mute too?
I think we're in pretty good shape. There
Yeah.
was a question about symbiotic relationships. Are these negative relationships or positive relationships? And I think you alluded to that. They can be either between the fungi and the arthropod.
Yes, so I'm trying to think of any symbiotic relationships between fungi and arthropods. Now, there are lots of, there are ants, the farm fungi, stuff like that. Things that live inside there are definitely fungi that yeasts and things like that help insects digest certain things. So those are beneficial. But in this case, when I was talking about, these are all things that infect insects. They're feeding on the insects and causing them to die. And so, or to be kind of maimed, like bad off. So those are specifically that when you see those, but fungi do have some interesting relationships with insects, and some are transmitted by insects, things like that. That might be a good subject for a future PPP. But how about this? I'll go through and in the interest of time, I'll just go through and answer through the chat some of these questions, and I'll let Mike have his time.
Sounds great. Thank you.
All right, thanks, Matt. And just one little thing to add to that, a couple of those entomopathogenic fungi have been commercialized, so you can actually, not a homeowner, but commercially, Bulgaria is available and Metarhizium as insect control, biocontrol.
Yes, they're actually they can be very good under the right conditions. They're actually using, I think it's a Bavaria for bed bug, killing bed bugs. And there has to be high humidity in certain times, but the spores can be kind of spread out very easily. And once they contact the insects, they start to do their work. And in there's certain conditions, they'll actually finish the work and kill the insects. So yes, they're good biological control and can be commercially available.
That's the coolest thing I've heard all day. All right, I decided to do things a little bit different. And instead of doing the currently active diseases, I will do bolos at the end, but I decided to work a case study. So hopefully folks are seeing are you seeing the single slide there of the
Yes,
case?
yes.
So this was an image sample that was submitted. The homeowner wasn't actually, the address wasn't actually in Caswell County, but the location from which it came was, so that was good. They put in where it was actually from October, just a couple of weeks ago. Didn't say what the rose cultivar was, but they checked off that they had yellowing, mottle patches, random distribution or patching distribution, and on the leaves. And the comments said, I am concerned this is mosaic virus. I received this plant in early September and it started showing these symptoms in the past week or so. And I just want, I'll go ahead and give you the pictures full screen here so you have a little bit easier time looking for things. I just want to have everyone take a moment and think about your process, your diagnostic process, and also your recommendation process. So what do you see here? Try and see as much as you can, and we'll see if we can call on a volunteer. Will there be a way that people's microphones can be turned on so that we could get one or two folks?
Yep, anybody could unmute if they have a comment. They can go ahead and unmute.
Well, let's do that after giving folks enough time to really kind of absorb what they're looking at here.
Okay.
So, Matt, weren't you going to sing while we were doing this?
No one wants that.
So we're getting some comments in the chat, Mike. Observations that it's not uniform, looks potentially like a virus.
All right. Keep
So far,
thinking. Mhm.
I think someone's making a note of the location of the symptoms. Susan was saying distant leaves. Maybe she means older leaves. I'm not sure. Susan, if you want to unmute, that'd be great.
No, I meant the I can't think of the scientific term, but the most furthest away from the stem.
Distal.
Oh, okay.
Distal,
is the
yes,
word.
distal.
Yeah.
Sorry.
Yeah, I would use distal in the sense of like farthest away from the base of the plant, but you could, I suppose, use it in, the distal leaflets on the stem. But okay.
I would, this is Tom Gale. I would be interested to know how long it's been since this plant was transplanted. I suspect strongly that it's a moisture or nutrient issue and therefore not spreading to all the plants because some of the root system may be okay, some not.
Well, this was planted in September, I believe it was.
And then this is October.
And this is right. Oops, I'm sorry. Back up. Giving things away here. Yes, plant received this plant early September and it started showing these symptoms in the past week or so, which would have been the middle to late October.
I think it's also bizarre that there's a little bit of yellowing on those two leaves that are closest in the front of the picture. At the very bottom of the leaves are.
The leaflets here. Can you see my cursor?
Yes, yes, exactly. Yes.
Mike, did we get any information about anything cultural that might have happened around this plant? Anything sprayed or any strange weather event? Any other information that we can have?
that was the whole of the information that we got was
Okay.
that sentence about the concern about mosaic virus.
Okay.
Someone just, Sandra wrote that there's some dark green leaves at the bottom that have black fungal spots. Good. So that brings up an important point. There appear to be more than one thing going on here. And I'll just we'll start the sort of reveal here. I don't have an actual title or anything to show it, but the folks who commented about the irregularity of the pattern, and let's see, what was the other comment there? Not uniform. That is a very good thing, and also the yellow color. So this is very typical of rose mosaic, in fact. And that's what I wrote them back that I believe it is, which turns out to be caused by one or, well, there are three viruses that can be involved, either alone or in combination. Let's see, it's prunus, necrotic leaf spot, apple mosaic, and Erebus mosaic. So we don't know exactly which one or ones are involved in this particular plant, but once you know that, what would be the conclusion? What's the implication of that? And I wanted to give you, there's a great reference, which is the Compendium of Rose Diseases and Pests from APS Press. Hopefully some of the offices, the county offices have this in print, because it appears to be one of the ones that is not available online through the libraries. A lot of those compendiums are, but this particular one, the rose one, does not appear to be. So does anyone know what the implication of this would be? Someone asked, could it be, someone asked about drainage and water. Someone asked, could it be the soil? Okay, well, we'll get there. I thought the virus that the plant needs to be destroyed. Dig it up, destroy. Out of here. All right. If that's, this turns out to be a case where the symptoms of the virus, the effects of the virus, are really pretty mild. Also, the chances of it spreading are very low. So I recommended not planting any other roses where they might have the roots intermingle with this one in case it formed a root graft, and you could possibly get the virus to pass through that way. But with this virus, and Jean is saying, I had 3 roses that have it for at least 2 years with minimal problems. So yeah, this is not one. Well, first of all, you're not going to cure it. So you're correct in that. So when you say it has to be destroyed, if you don't want virus yet, that's the only way to get rid of the virus, and to get rid of the plant. But in this particular case, the symptoms are pretty mild. So you may have reasons that you want to actually conserve this particular plant. You know, a particular flower, particular sentimental value, or maybe just the symptoms don't seem that unattractive to you. So the fact that it has rose mosaic is not something that would necessarily mean you have to destroy it. Now, obviously very different from rose rosette. I'm glad no one went there with that, because the symptoms are very different. The consequences are different. And of course you would want to remove and destroy it that way. But some other folks were pointing out some of the other things, and that's part of what I wanted to do with the exercise, was to have you notice, as someone did, that there's black spot on there. So this is someone who, if they've got a particularly sensitive cultivar, they're going to either have to have a high level of tolerance for some leaf spotting, yellowing, leaves dropping off, which can be pretty severe on some cultivars, or else they're going to have to get on a fungicide program next year when things start to leaf out. Someone else mentioned the soil, and that was a thing that caught my eye as well. This looks like there's kind of a hodgepodge of material there. There's some native soil, kind of the redder color there, and then looks like they may have top dressed with another soil or some kind of, it doesn't look exactly like mulch, maybe it was potting soil of some kind. So I would say that those who suggested a possible nutrient problem, they're also following a good line of reasoning there, where you may want to get the soil tested, because some of the general yellowing, not the blotchiness, but the general chlorosis there on the shrub does seem to suggest a soil problem. And the fourth thing would be, this thing is really leggy. So I just said to them basically to make sure and prune this when the time comes, in the late winter, early spring, when the leaves are just starting to open, so that they can get a better form on this. Let's see, Susan, I've seen those yellow leaves on my knockouts in combo with black spot and just thought it was part of black spot. Well, with black spot, you wouldn't see the yellowing without having the spots as well. So yellowing by itself will not be something that that black spot would do. Now let me, yep, I think that's about what I wanted to do with this. And okay, if you have other roses, would you recommend bleaching your pruners in between plants? Thanks, Gene, for asking that, because I forgot to mention this. Basically, the way that these viruses spread is through propagation. So if you're taking cuttings and rooting them, or you're a rose producer, that's how it's going to spread. It's not going to spread on pruners, nor is rose rosette, but there are some other things that could. So you definitely want to, if you're pruning your roses, you want to be sanitizing those periodically with, it could be diluted bleach. Well, of course, you have to be careful with the possible corrosion effects on your pruners there, or with alcohol. You got to make sure you have it in the alcohol long enough to have a sanitizing effect. But you do want to be doing cleaning of your pruners periodically during the process, but for other diseases, not for the viruses. So that's correct. This is not transmitted by insect or pruning tools, budding, grafting, etc. That is correct. But that includes possibly, I think I saw some reference to the fact that this has been shown to spread through root grafts. So if you had two roses right next to each other and they grafted the roots together, they themselves, the plants just naturally formed root grafts, you could get the virus passing that way. But it's really very low risk. Oh, and I think one of them is pollen transmitted too. But anyway, the bottom line, very low risk of spread from this individual to any other roses. All right. Let us move along to the bolos then. Somebody suggesting some pyrotechnics there. And I kind of spoiled the quiz here by clicking too hard on my mouse a moment ago. But first, I have two questions here among the bolos. The first one being, what do these two trees, there's an arborvitae there and a maple, what do they have in common? And I will launch a poll. So, do you think these two have nothing in common? Do they have natural senescence going on? Is it Ceridium canker or is it Verticillium wilt? It'll give folks a moment to think about what that is. And we've maintained good participation throughout here. I see that there's still 104 folks logged on, so that's good. People haven't gotten bored and dropped out. 38 seconds. We've been using kind of 40 seconds or 75% as our benchmark. We're lagging behind that 75. So we've got, I'm going to hold out and see if we can get to 50%. There we go, 50% participation. And so I'll end that and share the results. And the majority are in fact right here with, it is a natural senescence that we're seeing in both cases. What we've got is the fall leaf color, of course, that we all recognize on the maple, and something that's sometimes mistaken for a problem, a disease, or other kind of problem, which is the internal browning, the loss of those older needles on, especially arborvitae, but you may see it on Chamaecyparis, you may see it on some other evergreens. But it is also part of natural senescence, as I like to say. The tree may be evergreen, but that doesn't mean that every leaf and needle is immortal. So these will be shed at some time in the year, and this is the time when it's on arborvitae. So definitely be on the lookout for this happening. And in some cases, I see a little bit more than you would expect on a healthy tree. This is a particularly healthy tree, so you may see a little bit more extensive ones. Maybe there's some stress involved too, but very often this is not something that the homeowner needs to be worried about. So let's move on to our BOLO lists. Be on the lookout for these between November and when we next meet in late February. Well, every time of the year, it's something to be on the lookout for, be aware of, is Phytophthora and Armillaria root rots. Not necessarily that the organisms will be active, but that the decline, the symptoms can show up at any time of year. And there on the right is a section of a tree stem with the signs of Armillaria there under the bark mat of fungal mycelium. Phytophthora produces no such sign. We have to do a laboratory test for that. With privet, which since it's holding onto its leaves, people start noticing it more as other trees and shrubs lose theirs, but it's very common to see Cercospora leaf spot on privet. There are a couple different species, maybe even genera involved. Very typical large yellow blotches there on the privet. Again, not a major concern. Internal browning, as you mentioned in the quiz there, on arborvitae, fall cypress, and some other conifers. And one that's is an actual disease and kind of a more serious aesthetic problem would be Rhizosphaera needlecast on spruce. In fact, we just got a sample of that in yesterday at the clinic. Here's a picture of that. And one of the ways you recognize this is the needles are being affected individually. So it's not the whole branch, but just needle by needle, yes or no, it's turning brown, and then they will fall off. It happens in the lower portion of the tree especially. Continuing with trees and shrubs, of course, we have to mention boxwoods. So be aware of boxwood blight, not just in the landscape, but also potentially if you buy fresh boxwood wreaths or any holiday greenery that may be made from fresh boxwood cuttings. Those tips can contain actually the boxwood blight fungus. So you want to make sure that those are not, tossed out in the compost pile just in case. You want to make sure that you throw those in the regular trash at the end of the season. Maybe keep them away from where the rain might splash anything onto the boxwoods in the landscape and so forth. But just kind of inspect your greenery before it goes up. And then boxwood stem canker, as you see in the other photograph there, the individual branches with yellowing leaves, but the leaves hang on, they don't fall off. And of course, the below ground problems, so root rots like Phytophthora and nematodes, of which boxwoods suffer from at least three different kinds. Entomosporium leaf spot on Indian hawthorn and any photinias that may be left out there, you may see. So that's the lower center picture there with the bright red spots. And as we have mentioned in the program before, different cultivars vary a great deal in their susceptibility to or resistance to this particular fungus. Roses, Armillaria root rot, already mentioned before, be something that could take a rose bush out entirely. And rose rosette, we just have to be on the lookout for symptoms of that at any season. As leaves drop off, we may be more aware of black knot on some of our prunus species, particularly the plums, but also apricot and black cherry. You may see it, and that's just the lumpy, now this time of year, be black growth on the foliage, on the stems caused by infection by a fungus. And finally, multiple kinds of trees and shrubs may have these problems. Septibasidium, or the felt fungus, which can be different shades, so it can be very dark and black like this one. I think that's on Nissa. Other kinds may be a lighter tannish color, and that's an unusual one that it's not actually attacking the tree, but it's growing on colonies of scale insects that are on that stem. And this time of year, of course, wildlife damage, sapsuckers may be passing through and feeding on trees, and voles underneath the leaf litter, potentially feeding on some of the roots of your plants, not to be confused with moles. Moving to the flower garden, the flowers and perennials this time of year, of course, is the last gasp for the rust and the powdery mildews on a number of species, and maybe the first examples that we get of cold injury coming in. With Liriope, the tips with anthracnose, and that's why you want to cut those down closely in late winter so that we don't have too much fungus carrying over next year. So that's the photograph there in the upper left. But you can also have some Fusarium crown rot, and Phytophthora also can cause a crown rot or Liriope, but you'll see whole sections of the plant dying out instead of just the tips. And with the pansies going in for winter color, be careful to not plant infected material with either black root rot or some of the crown rots that they can get. Phytophthora crown rot is something to be on the lookout for, as well as Pythian root rot, which can come from the soil as well, and botrytis blight. So the lower right photograph there with the kind of matting foliage, and that will be more in the warmer late winter days. So we start to see that just generally in the environment. Quickly, our second BOLO quiz before we talk about the vegetables. Which of these photos shows black rot? And I guess we only have A and D. So you'll have to, if anyone answers D for this, then that would not be good because the only choices are A through C. And since time is of the essence here, I'm going to go ahead and end the poll quickly and share these results. So I know Charlotte's going to want to do her announcements. I see that most folks have answered B. Interesting. It's actually A. So these are in order. A is kale, B is, I think it was collards, and C is turnip. But black rot is typically, it doesn't always do this, but it often has those V-shaped lesions yellowing at first, and then at the edge of the leaf. That's a bacterial disease. So that's one of the ones you want to be careful about, making sure you have clean transplants going into the garden. B was actually a different disease, which we'll get to that here. So black rot, that's bacteria, Xanthomonas, that was on the kale. Sclerotinia stem rot is what you had on the collards there, and you can see some of the fungus there actually visible, the mycelium there, as well as the decaying main stem and petiole here. And then the turnips on the far right there, that is a disease called white spot, so a fungal leaf spot disease, Mycosphaerella. Cercospora leaf spot you may get on Swiss chard, beet, and spinach. So it'll be a relative of the white spot on turnip, but different hosts there. And of course, be on the lookout for your soil test results, because you've sent in your soil samples before the busy season fee kicks in. And of course, on top of your winter weeds and your seed catalogs, which will be coming to brighten your winter days. And for your turf grasses, real quickly, fairy ring, large patch, which would be on the warm season grasses when they're under stress in the cool weather, and yellow patch is another one that could be found on turf grass during the winter months. Finally, if on the long winter nights you want some reading material to help you get some sleep, our website does have the BOLOs month by month for most of the diseases, but Matt has added some of the insects, some of the pests there as well. And
Excellent.
I'll pass it to
Thank you. Thank you so
our
much, Matt and
leaders
Mike.
here.
That was a really fun episode with some really crazy things that we just didn't even know was out there. So looking closely is the message that we are taking away from today. As we all know, that this is the wrap up for our Plants, Pests, and Pathogens 2022 season. So just keep on the lookout for the follow-up email, which will be coming out to you soon. On that, we're going to have a survey just to get your feedback on this past season, what you liked, what you found most helpful, and what you might like to learn about in the future. this is the time that we get together and we plan for 2023. So please take a look and fill out that survey. It really helps us. And with that, I'm going to turn it back to Charlotte for our announcements.
All right, great. I just have a few announcements to close us out. Starting with, of course, and let me drop this in the chat. Our announcement slides are posted, so you can view them. You can actually download them. So if you ever see anything in the announcement slides you'd like to include at an upcoming meeting, or if it's something that is appropriate for the general public that you'd like to include in a class or event, you can go and download these slides and you're welcome to use them. So we're not doing plant these instead today with so much going on in our season wrap up, but we do want to remind you that all of our plant these instead slides are available in the archive where you can review them for both 2021 and 2022. And of course, these slides, as we said, you can download them so you can get all the links from the slides and from the link I shared before where you can view and download the slides. But when you go to the Plant These Instead archive, you can find all of the slides. You will see there are a lot of links on them, which will be the underlined blue text. And when you click on them, it will take you to either the plant toolbox where the full profile is for that plant or other references related to the feature. So I encourage you to go back and review those and use them to select better plants for your landscape. We had an excellent Extension Master Gardener College the week before last, and we are wrapping everything up or doing all of our follow-up, which includes we are working on a resource document that includes all of the resources that our different presenters shared. So that will be coming out very soon. It'll be posted on the EMG College website where anybody can go and see if a presenter shared their slides or if they recommended a website or if there was an extension publication related to what they talked about. You will be able to go and find those. It actually will be posted by the end of this week. Our next EMG College will be in 2024. So it is a every other year conference. So we will start talking about that at some point next year. But we do want everybody to know that the International Master Gardener College will be next year, 2023. That's one of the reasons we have planned for our North Carolina EMG College to be every other year to allow people the opportunity, if they want to attend the international, it's always in the odd years. We'll keep EMG College in the even years. So the International Master Gardener Conference is a wonderful opportunity to hear great speakers and interact with Master Gardener volunteers from all across the country and from Canada and South Korea. And I've heard potentially England was starting a program and they may have some folks there. And Overland Park, Kansas next year, June 18th through 22nd. So registration has just opened. It'll be open through March at the early bird rate, which I think is around 400. And then it will increase I think through the middle of April. So I encourage you to go to their website. They have an amazing website with all of their different speakers and features, just wonderful information, great pictures, really makes it look like, exciting, something you want to go to. So go check that out and start thinking about registering for the IMG Conference 2023. Of course, there'll also be a Search for Excellence, International Master Gardener Search for Excellence. I don't have the details on that yet, but typically they'll start asking for applications for that around January. So be thinking about any projects you've had in your county that you would like to submit for the International Master Gardener Search for Excellence. And those awards would be presented at the conference. If you are looking for other learning opportunities or thinking about Christmas gifts, the online plant ID courses, they're wrapping up for this year, but there'll be a whole new series advertised and available for next year. And wouldn't that make a great Christmas present for somebody to give them the gift of knowledge by purchasing the course for them and then they can take it. And this is relatively new. Now you can take them as an instructor-led course that's held over a specific period, or a self-study course where you can go in and do it, as you have time for a more extended period of time. So if you visit the site, the Extension Gardener portal, and you go to the online classes, you can find out all the information about how to purchase those and when the next upcoming sessions will be. You can also find there the information about the therapeutic horticulture courses, and those are offered in conjunction with the North Carolina Botanical Gardens. And there is the introduction to therapeutic horticulture course already planned starting January 2nd. So just as soon as the new year gets going. And I think there are 2 more courses now that are kind of a follow-up to that introduction, but the introduction is a requirement for the other 2 courses. So if you're interested in getting into therapeutic horticulture for 2023, this would be a great way to start. And of course, the J.C. Ralston Arboretum, which is part of NC State University, always has a wonderful lineup of online classes as well as in-person events there at the Arboretum in Raleigh. But their online classes include their plant lovers tours, the midweek with Mark, and in fact, the next one is tomorrow, where Mark Wethington, who's the director, will be talking about the night garden. And he even has another one planned for next week, and they've got things going on in December. So I encourage you to check those out and take advantage of those opportunities, especially if you're looking to get a few extra education hours as we get towards the end of the year for the Master Gardener program and for the calendar in general. Of course, if you're looking for those educational hour opportunities, you can always check out the recordings for Plants, Pests, and Pathogens. Remember, from this season, they are posted on the EMG intranet. But for previous seasons, you can find them on our YouTube playlist. The schedule, once we have the 2023 schedule ready, it will be posted online. But if you want to review the schedule from this year or any year, you can go and find it. And as Caroline mentioned, we will be sending out that evaluation. And we really want to hear from you, what you learned this year, how we can improve next year, and especially any topics or speakers you would like to hear from next year. So with that, we wish you all the best until we meet again. And that will be when our 2023 season launches on February 28th. So hoping everybody has a wonderful holiday season and winter, and look forward to seeing you later, well, early next year. And just wanted to say a shout out and thanks to Matt and Mike for all their wonderful knowledge they bring and share to us, to Caroline for her excellent job facilitating the webinars this year, and to all of you for joining us. Thank you.
And thanks to you, Charlotte, for the organizing.